Hello and welcome you all to my channel Omni Bioentrance, and today we will be discussing IIT JAM Biotechnology 2015 question paper. I will be discussing only the biology part. If you are new to my channel, please do like, share and subscribe. I have uploaded still previous year question papers of IIT JAM Biotechnology. You can refer to my channel playlist for the same. These are MCQ questions. Coming to the first one. Which of one of the following most accurately describe the process of natural selection? Selection of selection of one species over a competing species. Yes. Selection of individuals that successfully defend themselves against enemies. Yes. Selection of individuals that produce more than the average number of offspring. Yes. Selection of individuals that are more attractive to the opposite sex. Yes. So this is an MCQ question, but all the answers are correct. Then next one. Find regulation of the expression of identify the statement that is true of operons. Find regulation of the expression of an individual gene are made possible by operons, not individual genes. It is cluster of genes. So A is not correct. Only genes involved in carbohydrate metabolism are present in operons. No, for tryptophan operon, that is an amino acid, that is also present. Feedback inhibition of the biosynthesis of multiple enzymes by a single small molecule is made possible by operons. That is correct. If you see the case of lac operon, beta galactosidase breaks down lactose, and when lactose is depleted, the product, that is glucose, act as a feedback inhibitor, preventing further transcription of the operon. So C is correct. In the case of inducible operon, the inducer binds to the operator, not the operator but to the repressor. Then next one. Signaling pathways usually comprise of several intermediate steps that are arranged in the form of a cascade. What is the primary outcome of such an arrangement? It is amplification of the cellular response. If you take the GPCR, G protein coupled receptors, when the receptor binds to the ligand, the G alpha subunit gets separated from beta, and gamma subunits G alpha get converted from GDP bound form to GTP bound form. It will activate the next one, that is adenyl cyclase. Adenyl cyclase will result in the more production of CAMP. More CAMP means PAK, protein kinase A gets activated. So it is amplification of the cellular response. Next one. Which among the following contains oxygen-rich blood in the human vascular system? So right ventricle, it receives deoxygenated blood from the right atrium. The blood is pumped from the right ventricle into the pulmonary artery, which carries it to the lungs. From the lungs, it gets oxygenated, and the oxygenated blood returns to the heart through the pulmonary veins, which empty into the left atrium. From there, it flows into the left ventricle, which pumps it out to the rest of the body through the aorta. So they have asked which contains oxygen-rich blood. So it is 2 and 3, that is aorta and pulmonary vein. Then next one. Choose the option that lists the correct sequence of steps involved in gene therapy. Injection of expression vector into the patient. Wild type gene is inserted into expression vector. Wild type gene is isolated and cloned. That would be the first step. So C and D we will see, but A and B will be omitted. So R is the first step. Then comes wild type gene is inserted into expression vector Q. Q would be option D will be correct. R, Q, and then P, injection of expression vector into patient. Wild type gene is transcribed and translated in the patient. So it is RQPS. Answer is option. Then cephalin, a biological surfactant it is an ethanolamine phosphoglyceride. Answer is option B. Then next one. In a population growing according to the logistic growth model. So logistic population growth model, it shows that there is a gradual increase in the population at the beginning, then this is followed by a rapid growth, and eventually there will be decrease in the rapid growth once the population meets the carrying capacity. So if you see the equation here, that is dn by dt is equal to Rn 1 minus n by k. dn by dt is population growth rate. R is the intrinsic rate of increase, n is the population size, and k is the carrying capacity. So as the population size, that is n, approaches the carrying capacity, 1 minus n by k approaches 0, which means the growth rate also approaches 0. So the answer here will be option B, that is the per capita rate of increase approaches 0 as the population nears the carrying capacity. Answer is option B. Then which part of the genomic DNA contains the sequence corresponding to the 5' untranslated region? It is exon. Answer is option A. Then next one. Which one of the following is not true of RNA polymerase II? It requires a primer to initiate transcription. RNA polymerase II doesn't require a primer to initiate transcription. So A is the answer, whereas others are correct. Next one. Choose the option that shows the correct pairing of the cellular components with their corresponding function. Dynein is beating of flagella, P3. So B and C we will see. Q, desmosome. Desmosome is attachment of cells together. So B is correct out of B and C. Then R is endosome. It is a membrane vesicle. That's also correct. S is kinesin. That is movement of organelles. That is also correct. Then next one. And enzyme shows highest activity in the pH range 2 to 3. At pH 4 and pH 7, the enzyme exhibit 50%, 1% respectively of its highest activity. Which of the following states of an amino acid residue in the catalytic site is most responsible for its activity profile? So the enzyme's activity profile shows that it is active at low pH, whereas as it loses its activity as the pH increases. This indicates that the enzyme requires a protonated amino acid residue in its catalytic site. So we will see the case of aspartic acid. It is having a pKa of 3.5. That is, it is largely protonated at pH 2 to 3 and is deprotonated as the pH increases. And the protonated form of aspartic acid is what is required for the catalytic site. So answer here is option A. At 50%, at pH 4, it shows 50% activity, showing that about half of the residues are deprotonated, and at pH 7, it is having 1% activity, showing that most of the residues are deprotonated. So the answer here is option A. Then next one. Choose the option that shows the correct pairing of the diseases with their corresponding causative organism. Chagas disease is Trypanosoma cruzi, sleeping sickness is Trypanosoma gambiense, malaria is Plasmodium falciparum, and plague is Escherichia pestis. That gives the answer as option B, P4Q1R2S3. Then next one. Choose the option that shows the correct pairing of the products with their corresponding microorganism. Citric acid. Citric acid is Aspergillus niger, P3. Polyhydroxyalkanoates is Ralstonia eutropha, Q4. Then gentamicin is Micromonospora purpurea and ethanol is Cybomonas mobilis. So answer is option C, P3Q4R1S2. Then next one. Determine the correctness or otherwise of the following assertion and reason. B cells secrete antibodies against a virus while cytotoxic T cells kill virus infected cells. That is correct. B cells confer active immunity while cytotoxic T cells confer passive immunity. That is wrong. B cells confer active immunity, okay, but cytotoxic T cells also confer active immunity. What is passive immunity? It involves the transfer of preformed antibodies or immune cells from one person to another. That is from the mother to the fetus. It is passive immunity. It only provides a temporary protection and it doesn't require that the recipient immune system mount a response. So A is true, R is false. Answer is option C. Then next one. Which one of the following option shows the correct pairing of the enzyme with its corresponding application? Papain, meat tenderizer, P4. Bromelian, gluten complex reduction. So bromelian, it is a mixture of proteolytic enzymes found in pineapple and it breaks down glutens by reducing its complexity and making it less likely to trigger an immune response in individuals with gluten related disorders. Then peroxidase is for immune assay marker enzyme and beta amylase is for maltose syrup preparation. So the answer here is option A, P4, Q1, R2, S3. Then coming to multiple select questions, MSQs. In a large wildflower population, assume that no new mutations occur and that no natural selection operates. So it is in Hardy-Weinberg equilibrium. What factors will affect the frequency of a genotype in this population? Non-random mating. Yes, Hardy-Weinberg is for random mating. Gene flow, in Hardy-Weinberg there is no gene flow, so gene flow will affect genotype. Frequency of a genotype. Outbreeding within a population also affect the frequency of a genotype. Invasion of a new pathogen that kills a large number of individuals in the population will also affect the frequency of a genotype. So A, B, C, D are correct. Next one. What is or are the difference between microtubules and microfilaments? Microtubules are made of tubulin and microfilaments are made of intermediate filament. How can microfilaments be made of intermediate filaments? Microfilaments are actin filaments. So A is not correct. Then microtubules are important for compression resistance and microfilaments bear tension. B is correct. Microtubules are important for the function of cilia and flagella and microfilament are important for cytoplasmic streaming. That is also correct. Microtubules muscle contraction. No. Microfilament ciliary movement. So D is not correct. Microtubules is for ciliary movement and microfilament is for muscle contraction. Then next one. N and N0 represent the number of viable cells at time t during sterilization and at start of sterilization t is equal to 0 respectively. Assuming that cell death follows first order kinetics and that k is the death rate constant, which of the following relationship is or are correct? So the formula is as given in option B, that is minus ln n by n0 is equal to kt, whereas option C is for second order kinetics. Answer is option B. Then next one. Which of the following statements about antigen-antibody complexes is or are true? Hydrogen bonds and van der Waals forces participate in antigen-antibody interaction. Hydrogen bonds and van der Waals forces are non-covalent interactions and antigen-antibody interactions involve non-covalent interactions. So A is correct. Ionic bonds and hydrophobic bonds participate in antigen-antibody interaction. Ionic bonds and hydrophobic bonds are also non-covalent bonds. So they are also correct. The combined strength of all the interactions between a single antigen binding site on an antibody and a single epitope is called avidity. It is not avidity, it is affinity. If it was avidity, then it will be multiple antigen binding sites on an antibody and multiple epitopes of the antigen. So C is incorrect. Sorry. Antibody elicited by one antigen can cross-react with an unrelated antigen. D is correct. So A, B, D is the answer. Coming to numerical answer type questions. An enzyme preparation containing 10 mg per ml protein shows a specific activity of 50 units per mg. The initial velocity of the reaction in a standard 1 ml reaction mixture containing 10 microliter of the preparation in micromole per minute per ml is. So you have to calculate initial velocity. So for that, first you have to calculate the amount of enzyme in 10 microliter reaction mixture. So 10 in 10 microliter into 10 mg per ml will give you 0.1 mg of protein. Now calculate the total units of the enzyme in the reaction mixture. So 0.1 mg of the protein into 50 units per mg, that is 5 units. Since the reaction mixture is in 1 ml, the enzyme concentration is 5 units per 1 ml or 5 units per ml. The specific activity is defined as units per milligram of protein. So 5 units per ml is equal to 5 micromole per ml per minute, since 1 unit is 1 micromole per minute. So initial velocity is 5 micromole per ml per minute. Answer is 5. Then next one. Assume that the gene UNC is essential for the coordinated movement of a nematode and that the wild type allele capital U is dominant over the mutant allele small u. Similarly, the wild type allele capital D of another gene DPY, which is responsible for the normal body length, is dominant over the mutant allele small d. Assume the UNC and DPY are on two different chromosomes. If a female of genotype capital U, capital U, capital D mates with a male of the genotype small u, small u, small d, the percentage of the F2 progeny that will display uncoordinated movement but will have normal body length is. So what is it? They have given UNC, U dominant and it shows coordinated movement. Small u is recessive and it shows uncoordinated movement. Capital D is dominant, normal body length, and small d is recessive for short body length. Now female homozygous for U, capital D is crossed with male homozygous for small u, small d. These are the gametes. This is the F1. F1 individual show coordinated movement, normal body length. F2 is obtained by crossing the F1 with itself, selfing. These are the gametes. So this is the Punnett square. These are the 16 progenies that are obtained. They have asked for the one which is showing uncoordinated movement and will have normal body length. So uncoordinated movement is recessive character. So it should be small u, small u. And normal body length can be capital D and capital D small d. So that is seen in 3 cases, 1, 2 and 3 out of 16. So the ratio will be 3 by 16 into 100, so 18.75%. The next one. The pH of gastric juice in the stomach is 2. However, the pH inside the cells that line the stomach is 7. So gastric juice is 2 and the cells that line the stomach is 7. For transport of protons from inside of the cell to the stomach, the free energy change in kilojoule per mole at 37 degrees Celsius is. So delta G is given by the formula minus RT ln K. R is the gas constant. They have given it as 8.314. T is temperature at 37 degrees Celsius. So convert it into Kelvin. You will get it as 273 plus 37. That's 310 Kelvin. K is the equilibrium constant and it is related to pH gradient. So to calculate K, you have to subtract pH inside the cell minus pH in the stomach. So 7 minus 2, that is 5. K is equal to 10 raised to minus delta pH, that is 10 raised to minus 5. Delta G is equal to minus 8.314 into 310 into ln 10 raised to minus y. You will get it around 29. So that will be the value of delta G upon calculation. Then next one. Drosophila melanogaster is a diploid organism having 8 chromosomes. The number of combination of chromosomes which are possible in its gamete. So there are 4 pairs of chromosome and the gametes will receive half the total number of chromosomes. So each gamete will be having 4 chromosomes. And the possible combination of chromosomes in a gamete can be calculated using the formula 2 raised to n, where n is the number of chromosome pairs. Here it is 4 pairs. So 2 raised to 4, that will be 16. Then next one. The deactivation rate constant of an enzyme is 0.346 per hour. Assuming that the deactivation process follows first order kinetics, the half-life of the enzyme in minutes is. So K is 0.346 per hour. Convert it into minutes. So divide by 60, you will get this value. Then use the formula T half is equal to 0.693 by K. Substitute the values, you will get it as 120 minutes. Then next one. A solution containing NAD plus and NADH has an optical density of 0.233 at 340 nanometers and 1 at 260 nanometers. So solution is having optical density of 0.233 and 1 at 340 and 260 nanometers. While this solution absorbs at 260 nanometers, NADH alone absorbs at 340 nanometers. Okay, so NADH absorbs at 340 nanometers. All measurements are carried out in a 1 centimeter cuvette. Given the extinction coefficients in the table, the concentration of the oxidized form of the cofactor in micromolar is. So we know the Beer-Lambert's law, A is equal to epsilon Cl, where epsilon is the molar extinction coefficients. So at 340 nanometers, absorbance is equal to, it is of the solution. So we have to take into consideration both NAD plus and NADH. They have asked for the oxidized form of the cofactor, that is NAD plus concentration. So 0 plus 6220 into C2, where C2 is the concentration of NADH. You will get it as 3.74 into 10 raised to minus 5. Then at 260 nanometer, absorbance is 1. Again use the same formula. 18,000 into CNAD. You don't know the concentration of NAD, but you know the concentration of NADH. And 15,000 into 3.74 into 10 raised to minus 5. So CNAD plus will be 24.4 micromolar after calculation. So that's it. Thank you for watching my channel. Please do like, share and subscribe. If you are new to my channel, please do subscribe it. Then I have uploaded previous year question papers of IIT JAM Biotechnology. You can watch them in my channel playlist. Thank you.